and I'm going to continue on the histology of the central nervous system. So we had completed the spinal cord, and now we are on the brainstem. So at the medulla, the upper region, we have the fourth ventricle where it closes, and remember, it has to become narrow. We have the fourth ventricle in the medulla, but it narrows down to become the central canal of the spinal cord. So the medulla is divided into the upper part that is open medulla, where you have the fourth ventricle, and the lower part, which is the closed medulla, where it narrows into the fourth ventricle narrows into the central canal of spinal cord. So at the upper medulla is where you have the inferior olivary nucleus, and this is usually convoluted. So when you see a convoluted structure, it's the inferior olivary nucleus at the upper medulla. Then on its dorsal aspect, you'll see the dorsal accessory olivary nuclei, and on its medial aspect, you see a medial accessory olivary nucleus. What's the main function of the olivary nucleus? It helps to coordinate motor aspects because it gives projections to the cerebellum. Okay, so then in the medulla, we also have gracile and cuneate nuclei. Okay, as well as the gracile, fasciculus gracilis and fasciculus cuneatus. Remember, the fasciculi are carrying ascending fibers for discriminatory touch, vibration, and proprioception, and these terminate in the gracile and cuneate nuclei. And we said cuneate is carrying information from T6 and above, gracile is carrying from the lower limbs. Then at the medulla, we also have pyramidal decussation. So all these are occurring at the level of the medulla. Anything convoluted, we say, is the inferior olivary nucleus. Anything dorsal to it is a dorsal olivary nucleus, and ventral to it is a medial. Then we've said posteriorly we have fasciculus gracilis and fasciculus cuneatus, and high above at the medulla you'll have nucleus gracilis and nucleus cuneatus. Okay. Then this is the spinal nerve and tract of trigeminal nerve. These are the pyramids here. Okay, ventrally. This is the fourth ventricle. These are the nuclei of the hypoglossal nerve on the floor of the fourth ventricle. So this just shows a cross section of the medulla. So this is upper to mid level. Okay, the medulla. So how do you know this is lower part of the medulla and this is upper part? You can appreciate the ventricle, fourth ventricle here. But as you go down, the ventricle narrows because we need it to form the central canal of the spinal cord. So those are the main features. So upper medulla, you appreciate the convolution of the inferior olivary nucleus. And it's at the lower portion that you appreciate the fasciculi. Okay? So, remember these pyramids, they are carrying corticospinal tracts. So if you're to take a cross section of the pons, the pons has two parts, a ventral and a dorsal part. The ventral part is called the basal pons. It contains pons, also has descending fibers of two types. We have corticospinal fibers that are passing through ventral pons, and we also have corticopontocerebellar fibers. These are from the cerebral cortex to the pons, then later on they will end up in the cerebellum through the middle cerebellar peduncle. Okay? Then the dorsum part of the pons is called the tegmentum, and it mainly contains spinothalamic tracts that are carrying pain and temperature information, and also contain the nuclei of trigeminal, abducens, and facial nerves. We also have in the dorsal tegmentum of the pons, we have the medial lemniscus. Remember, medial lemniscus is just the dorsal column tract, carrying proprioception, vibration, and discriminatory touch, okay, through the fasciculus gracilis, fasciculus cuneatus. So all that information from the spinal cord to the medulla, then they will pass through after reaching the gracile and cuneate nuclei of the medulla, they now decussate, and that's what you call the medial lemniscus. So this medial lemniscus is seen in the dorsal tegmentum of the pons. So if you're to look at this histological section, this is the very discriminatory terminalis and nucleus cuneatus, then the decussation occurs and the lemniscus passes at this point. Okay. Then we move to the next slide. Coordinate the muscular activity and to maintain posture and equilibrium. There's a lecture on the cerebellum, so you can go and check in detail the functions. So the cerebellum has an outer cortex, which is the gray matter, and an inner white matter. the white matter contains axons of neurons, but within this white matter, we still have 4 pairs of nuclei. Nuclei are a collection of neuronal cell bodies, and the arrangement of these nuclei include the dentate, emboliform, globose, and fastigial nuclei from lateral to medial. Then the cerebellum, within the cerebellum, you have nerves that will communicate the cerebellum with the other 3 portions of the brainstem. So the cerebellum is able to communicate with the midbrain via superior cerebellar peduncle. Cerebella communicates with the pons via the middle cerebellar peduncle through the corticopontocerebellar fibers via the inferior cerebellar peduncle. The cerebellar cortex usually has convolutions or folds, and these are what you call folia. So you can see the folds, these are folia, the branching central medulla of white matter. So this light blue is the cortex. The cortex of the cerebellum is divided into 3. So you have outer molecular followed by Purkinje, then the inner granular layer. So outer molecular has few neurons that are unmyelinated. Then the Purkinje is the middle layer, and these Purkinje cells, they are so this is the molecular layer, this lighter region. Then Purkinje layer is just a single layer of flask-shaped cell bodies of neurons. So Purkinje cells have flask-shaped cell bodies, and then their dendrites project into molecular and granular layer. Then granular layer is called granular because it resembles sand granules. So Purkinje cells, how do you describe them? They are large cells. Cell bodies are large and flask-shaped, and they have a fine axon that extends to the granular layer and dendrites arborizing into the molecular layer. So this is molecular layer has neuronal cell bodies. Purkinje layer, you're able to see large flask-shaped cell bodies with dendrites getting into the molecular layer. Then the granular layer, they resemble granules of sand. Okay, so you have the granular cells within the granular layer. So Purkinje, these are Purkinje cells. You can see the arborization of the dendrites into the molecular layer. Look at the flask-shaped cell bodies and the dendrites arborizing into the molecular layer. Then we have stellate and basket cells in the molecular layer. So molecular layer has stellate and basket cells. Purkinje layer has Purkinje cells. And then the granular layer has granular cells and Golgi cells. So the clinical correlates for the cerebellum, you have cerebellar ataxia, which can occur where you have damage to the Purkinje cells, and that affects coordination of the voluntary movement. And this can be caused by neurodegenerative diseases, alcohol abuse, tumors, trauma, hypoxic damage. So all this can lead to cerebellar ataxia. Then we go to the midbrain, histology of the midbrain. So the midbrain has the dorsal aspect, it's divided into 2, yeah, anterior tegmentum and posterior tectum, and the cerebral aqueduct of Sylvius is sort of the landmark. So anything posterior to aqueduct is the tectum, and anteriorly is the tegmentum. So tectum is dorsal to the central gray matter, and the central gray matter is around the periaqueductal gray, and it's made up of superior colliculi above and inferior colliculi below. So 4 of them, 2 superior, 2 inferior, they form the quadrigeminal plate. Then anterior to the central gray matter, we have the tegmentum, and this usually contains ascending, and so this is the tegmentum. So the midbrain has cerebral aqueduct. The aqueduct is a tectum, anteriorly collicular. So the inferior colliculi allow auditory reflexes, or rather they are responsible for auditory reflexes. Then anterior to the cerebral aqueduct of Sylvius is the tegmentum, and the tegmentum has tracts. Okay, so you have lemniscus carrying proprioception, discriminatory touch and vibration. You have trigeminal lemniscus from the head and neck, spinal thalamic tract carrying pain and temperature. You have the lateral lemniscus, the rubrospinal tract. Then you have different nuclei within the tegmentum, mesencephalic nuclei of trigeminal, nuclei of trochlear, dorsal and ventral tegmental nuclei. Then lastly, the basal part of the midbrain, anteriorly where you have cerebral peduncles and the substantia nigra. So that's what you see at the cross section of at the inferior colliculus level. Okay. Then, so if this is at the level of the inferior colliculus, you'll be able to appreciate the 4th cranial nerve and its nuclei. the inferior colliculi posteriorly, cerebral aqueduct, periaqueductal gray matter with a nucleus of trochlear nerve, and trochlear nerve is seen emerging from the nucleus, and it goes dorsally, and they cross, the right and left cross. Then there's tegmentum with the tracts and the nuclei, and there is cerebral peduncle and the substantia nigra. So those are the structures that are seen at cross-section of the, you can be asked, draw the cross-section of the midbrain at the level of inferior colliculus, showing the structures present. So within the cerebral peduncle, remember to show corticopontine fibers anteriorly, corticopontine fibers posteriorly, but the middle portion has corticospinal and corticobulbar fibers. At the level of superior colliculus, you still have cerebral aqueduct of Sylvius, periaqueductal gray matter, but of note in the periaqueductal gray matter, you have nucleus, motor nucleus of oculomotor, as well as the Edinger-Westphal nucleus, which is the parasympathetic nucleus of oculomotor. Then you'll see oculomotor nerve emerging. Posterior to the cerebral aqueduct of Sylvius, you see the superior colliculi. Anteriorly, you see the tegmentum with the tracts and the nuclei. Then you appreciate the cerebral peduncle with corticopontine fibers, anterior and posterior, and corticospinal and corticobulbar fibers at the center. Then behind the cerebral peduncle, you see the substantia nigra and the red nucleus. So the difference between cross-section of the midbrain at the inferior and the superior colliculi at the colliculi, then the nuclei of trochlea at the inferior collicular level, and the nuclei of oculomotor, the motor nuclei and Edinger-Westphal at the level of superior colliculus. Then you're able to appreciate the red nucleus at the level of superior colliculus and not at the inferior colliculus. So when you're asked to draw a cross-section, those are the things to check for. Substantia nigra has two parts. The zona compacta is posterior. It has lots of melanin, while the ventral part is the zona reticulata that has ion compounds. So substantia nigra is part of the basal ganglia, and it has connections with the cerebral cortex and the spinal cord and the corpus striatum, and it helps to control motor function. It's basal ganglia, and usually it contains dopaminergic fibers, so it uses dopamine as the neurotransmitter, and dopamine is inhibitory on the corpus striatum. Remember, the corpus striatum has the caudate, putamen, globus pallidus. So usually at birth, there's less neuromelanin, okay, within the zona compacta. But as you age, the melanin increases. So this, we've said the dorsal is zona compacta and anterior is zona reticulata. So this is zona compacta, zona reticulata. So that's the substantia nigra, okay? And you can appreciate the melanin pigments when you do a histological section. So this is a cross-section of the midbrain at the level of, how do you know? Presence of red nucleus tells you this is the level of superior colliculus. So this will be superior colliculus, this will be nucleus of oculomotor. Oculomotor nerve goes ventrally, it pierces the red nucleus. While if it was at the level of inferior colliculus, the trochlear nerve leaves trochlear nucleus, and then it courses dorsally. Okay, then these are corticopontine fibers, corticospinal and corticobulbar fibers, substantia nigra, red nucleus. The medial lemniscus is here. So these are the tectum, and this is the tegmentum. So clinical correlates of substantia nigra is Parkinson's disease. It's a neurodegenerative disease characterized by tremor, rigidity of muscles, akinesia, which is slow movement, and postural defects, TRAP. And it's because of degeneration of substantia nigra, so the nigrostriatal pathway, nigrostriatal nerves are degenerated, dopaminergic nerves are degenerated. So the substantia nigra looks pale, it loses the pigment, and how do you treat this? You give L-DOPA. You cannot give dopamine because dopamine will not cross the blood-brain barrier. So you give L-DOPA, crosses the blood-brain barrier, then it goes and raises the brain dopamine levels. So you're able to control the symptoms of Parkinson's disease. So that just shows you how you lose the pigmentation